All right, so I will introduce our next speaker. He was in fact introduced in my talk, Kuba Filipkowski. Kuba Filipkowski is a behavioral neuroscientist with extensive background in molecular and cellular biology, including studies of transgenic animals and gene expression. And right now he's the head of behavior and metabolism research laboratory at Mossakowski Medical Research Institute, Polish Academy of Science. Kuba, the floor is yours.
Hi, everyone. Thank you for your kind introduction, and to both Pavel and Adam for inviting me to join the organizers. Can you hear me, people?
Yes, we do.
Good, good. Thank you. And I'm going to talk about ultrasonic playback experiments that we're doing. Here's the list of authors. I need to start with it because I tend to forget about acknowledgements, and I really need to acknowledge Krzysztof, Rafał, and Agnieszka, who mainly worked on the results that I'm presenting today, and all the authors are here. We thank also funding from NSC. Markus Vor mentioned today that the deep understanding of natural behaviors of rodents is crucial when we think about modeling, for example, human diseases and investigating them in rats. This is a picture from a recent review in eLife by Schweidpurf, and who reminds us that rats are group animals. They live in colonies. They are active at night, and a lot of things are happening in these colonies where rats huddle and sleep together, where males approach females without success and successfully, and then females give birth to pups. Also, fights happen within the colony, but mostly outside the colony with strangers. But the dominating behaviors within the colony are amicable behaviors, including, for example, grooming. Most of our studies, most of the studies concerning ultrasonic vocalizations tend to focus on interactions, and on interactions happening in close contact, like copulation or fights that I mentioned. However, when we think about the corridors which are underground, which are dark, so animals can't really see each other, the ultrasonic vocalizations give theoretically a good means for communication. If we move that rat into this corridor that I emptied a little bit, and we introduce it to the intruder in the colony, or rat within the colony, they might interact with each other using ultrasonic vocalizations. In fact, it was proposed in 2010 by Stefan Brudzyński and Neville Fletcher that ultrasonic vocalizations are ideal for short-range communications within underground burrows due to several physical features of ultrasounds of vocalizations, of these vocalizations. So thinking about it, we thought that it's worth trying to find how this communication may occur, and we focused on this communication, on this meeting of two rats, with one vocalizing, but we made it simpler to begin with. We replaced one of the animals with a speaker, and projected from a speaker four main signals to begin with. These are 50 kilohertz ultrasonic vocalizations, 22 kilohertz ultrasonic vocalizations, we built 10-second long signals like that, and we also manufactured artificial tones mimicking those to see whether there might be a difference in recognition. We're not the first to start playback experiments. I need to mention Markus Bohr's achievements in here. However, what we noticed and what I noticed when comparing with our results, that in his group's results, these investigators are seeing mostly 22 kilohertz calls as a result, as an answer, and very rarely appetitive calls of 50 kilohertz. We speculate that it might be due to a little anxiety involving or evoking setup with elevated plasmas or elevated open fields that are being used. We want to do some approach which is more home cage like in our endeavors. And we, as I was saying, we comparing 50 kilohertz playback with 22 kilohertz playback. Both exposures to these two kinds of ultrasonic vocalizations result in different reactions within the rats. We wanted to see what's the reaction to these different types of calls, and especially focused on the heart rate, which is known to be impacted differently by appetitive and aversive stimulation. And also, due to some theories, there is a connection between cardiovascular parameters and ultrasonic emissions due to polyvagal theory, polyvagal connection. So the first aim of our study was to evaluate the changes in behavior, heart rate, and ultrasonic emission after playback of ultrasounds. This was done in Wistar rats, which were 7 weeks old males, and we also compared paired and single rats. These were habituated to the situation, properly handled, and then there was done a surgery with implementation of radiotelemetric transmitters from DSI. After that, there was a recovery, during which again we have a habituation to the home cage like a setup and speaker outside of the colony, and the very experimental day with ultrasonic presentation when rats had at that point 12 weeks of age. We used playback of natural 50 kilohertz and 22 kilohertz ultrasonic vocalizations and analogous tone representations of those. These were given in packs of 4, intermingled. So we have different groups, 2 different groups in both paired and single rats. We changed the order of the presented sounds. That's what we always do. So we have counterbalanced groups in this regard. What we do first is there is a 10-minute habituation period which rats spend in the cage that they're accustomed to, and we measured distance, time spent in the half with the where the speaker is, heart rate itself, and number ultrasonic vocalizations. These are the results from the 2 groups that we investigated first. We noticed that heart rate levels, they gradually go down during this time, and they actually gradually go down throughout the whole experimental session. Moreover, single rats had a higher heart rate levels during most of the time as well. Also, we noticed that single rats vocalize more frequently. They vocalize more than paired rats. That's all what happened during the habituation session before the playback session itself, before the playback started. I'll try to show you. Okay, here's what happens during the session. When you can see the playback is off now, then the playback of 50 kilohertz is started, like now. The rat is waking up and starts to vocalize. Here's the count, the number of vocalizations. The playback is off now, but the rat is still vocalizing, still approaching the speaker that can be seen here. This is our very simple setup with home-like cage, rat accustomed to it, microphone just above it, and the speaker next to it. And we also, when we analyze this, we again measure these four values that I mentioned before, and present them in such a way. It's pretty complicated, so I'll try to guide you through the method we presenting our data. Here we have the first graph, and you can see that the time passes at a certain point that we mark 0. It's a 10-second long signal playback, ultrasonic playback, that was demonstrated on the video before, which lasts only 10 seconds, and then you have the time afterwards. We can measure different things. First, one thing we measure is the distance travel. The information you can see above. So the rats travel at some point, and during presentation of ultrasonic vocalization, they might change the distance they travel. On the right hand side, we have % of time spent in the half of the cage that has the speaker next to it. So the chance level is marked here by the dotted line, and we observe what happens when we start the playback, and we can see, like in here, that the rats on average approach the speaker. Also, we investigate heart rate, HR, and the number of ultrasonic vocalizations. Again, the low number of ultrasonic vocalizations before the playback, and abrupt, we can see it here, and rise in the number of such vocalizations when the playback is emitted, which gradually goes down. And we can also measure the heart rate, which is on the left side, in beats per minute. And we can see in here a rise in heart rate after ultrasonic playback. Here are the results that, of our first experiment when we given 50 kilohertz playback. Here you have the results from paired and single animals. Those are playbacks that are natural. Here are tone playbacks. What you can observe several things. We can observe that 50 kilohertz tone playback evokes a short, rapid increase in movement, and also which results in approach of the speaker. And we see the increase, significant increase in heart rate, as well as significant increase in the number of ultrasonic vocalizations. I like to stress this increase, that it's really for the first time, I think, we've seen such a high amount of vocalizations in response to playback, which is more than 60 vocalizations per minute. And that's also important because some rats vocalize really at low levels, and within those rats, one didn't vocalize at all. So there are rats that really respond to such vocalizations. So we think that we tapped into something real, something important, something behaviorally relevant. Here's the another part when the playback is in 22 kilohertz. The results are different. We observe actually not the rise in activity, but lower activity immediately after the cessation of the signal. We also observe the approach to the speaker, which is contrary to some other results observed in the literature, and we observe lowering of heart rate following aversive playback. This is all been shown to be significant. When you compare ultrasonic playback and tone playback, we don't see much difference. In general, both playbacks had similar effects, both in 50 kilohertz playback and in 22 kilohertz playback. Here we compare the results, the effects of 50 kilohertz playback versus 22 kilohertz playback. So we try to see what's the difference in impact of appetitive versus aversive playback. And what we see is the difference in behavior, that after appetitive playback, rats start to move, and rats stop to move after the aversive playback. And we see the big difference in the number of vocalizations and a big difference, big impact on the heart rate. The same is with the tone playback, similar results for the tone playback. When we combine these results, we see a really big significance of those changes that I described. There is some difference between also regarding approach. So what I was trying to show you, and I hope I convinced you, is that in our setup, we show that 50 kilohertz playback results in increasing heart rate, while 22 kilohertz playback causes decrease in heart rate. There are many conclusions, and we have hard time with reviewers all the time that we have too many results. And apart from those two main results I mentioned, I just remind you that rats vocalize really abundantly when exposed to recorded ultrasonic playback. So these are our conclusions and main findings, and these results have been published already. They are in iScience last year's paper. Another project that I would like to share with you is the one that we do more or less the same thing, but the rats that we were using were fear conditioned before. We wanted to have them a little bit more stressed before the playback, and honestly, the idea was that these rats will vocalize less after exposure to playback. They underwent fear conditioning at different levels of conditioning, with 1, 6, or 10 stress sessions, and and we did the ultrasonic presentation and fear testing, fear conditioning testing afterwards. What happened is that we tested them also for the between groups. We checked whether these group at the beginning show the same level of heart rate and, for example, ultrasonic presentation. It turned out that 2 rats were off, were outliers. We removed them from the analysis, but we actually calculated everything with and without them, and the main findings stand for both situations. We showed that there was increase in freezing in the fear conditioned groups. We also tested them in the 10 minutes habituation period, where the important findings were that the one trial group had lowered heart rate levels, and all fear conditioned animals vocalized more than control animals. That was first indication that our main hypothesis might not be good. Here I'm showing the heart rate increase and all the results that we observed for V-star rats before without fear conditioning. So we definitely have luxury of our reproducibility of our results, and the crisis that Pavel mentioned don't affect us that much, luckily. Here, however, we don't see the increase in HR in one trial group, which is quite often quite different. That's repeated result also, and there is a clear increase in activity in the groups following 50 kilohertz playback, and approach behavior that I'm showing now, increase in vocalizations, and increase in heart rate. As far as 22 kilohertz playback is concerned, we saw the drop in activity again, approach behavior, surprisingly, and both the decrease in heart rate here and here. When we compare 50 and 22 kilohertz results, we see again big differences that we've observed for the control animals. But what the key finding is that when we compare control animals, 0 trial animals, with combinations of animals that have been fear conditioned, we see that the conditioned animals, they move more, their locomotor activity is higher. Also, when they stop moving after 22 kilohertz playback, this difference is also bigger in fear conditioned animals. And what's most important for us, we think, is that the number of vocalizations is higher in fear conditioned animals. This is the most interesting finding in this second group of our findings of the second project. These results were recently published in Brain Sciences. Here are the results, the authors and the title. In our third project, which is incomplete yet, and we in process of analyzing data, we added SHR. These are rats that are spontaneously hypertensive, which are model for ADHD and schizophrenia. So we, on the top of the things that happened before, we added those rats. And I will skip some introduction and just highlight the many things. Here, the SHR rats had way lower, I'm sorry, they had higher HR heart rate levels than Vista rats. And regarding heart rate, when especially when you see comparison between 22 kilohertz signal and 52 kilohertz signal, we don't observe the increase in HR that's seen in the control animals, but we see a huge drop in HR, in heart rate, following 22 kilohertz playback. Also, what we observe is the much lower number of vocalizations in these animals. Here's the comparison between SHR and Vista rats, and we see differences in the number of vocalizations I marked here, which are way higher in Vista rats and much smaller in SHR. Conclusion for this part is conclusions are that SHR show higher levels of anxiety than Vista rats at the beginning. I didn't elaborate on that. And when during playback experiments, SHR, they vocalized less frequently, and the changes in HR were absent after 50 kilohertz playback and more profound after 22 kilohertz playback. These observations and a lot of others that I didn't have time to show will hopefully be the first description of SHR vocalizations. Take-home message: when exposed to the ultrasonic playback, heart rate of rats increases if the playback is appetitive, and heart rate decreases when the playback is aversive. When going back to our short-range communication thing that we hope, I think, takes place in the dark burrows of our cities, we think that when there is a signal from one rat, which might be appetitive or aversive, 22 kilohertz, there is a response from the other rat that is more copious in case of appetitive signal and less numerous when the signal was aversive. This magnifies when the rat is single or a rat is more stressed. This response is more evident, more numerous in both cases. Also, we observed differences in the behavior regarding approach and regarding activity. I would like to mention, as I was signaling it last time when I had a talk 2 years ago, that we have also other projects in the lab. Rafał Polowy is making a huge data set on how playback is happening when the rat is exposed to different kinds of signal, with different number of signals, with different interval of the signal. And Agnieszka Wardak had a very difficult task to find out whether it's possible to teach rats to vocalize, to evoke it with food reward, to reward vocalizations or answers to vocalizations, and it didn't work at all. So maybe there is something to talk about and share, and maybe publish these negative results as well. And I thank you very much for your attention.
Thank you, Kuba. We are a little bit short on time, so maybe just
we can skip questions.
One question, if there's any.
Yeah, I've got one question, if I can. Do you think it's those pre-shocking in the pre-shocking experiment? Do you think it's a matter of the arousal a little bit when you get the arousal after the shock, and then you get the exposition to the 50 kilohertz? Do you think this is the reason, the level of arousal state?
We don't have precise data to pinpoint what facet of behavior is being turned on here or changed here. Arousal is a perfect idea. Probably yes, and it would be interesting to to dissect what kinds of things we can do here, and try to see what exactly is the effect, or exactly is the cause of the difference. Thank you.
Thank you very much. All right, so